Apologies for that. Normal service will be resumed. Uh, so, um, yeah, so let me just get the agenda up again. I'm going to start off with a bit of a brief overview, and then I'm going to be passing over to Professor Yongju Huang of the University of Hertfordshire, who's one of the leading experts in the country on light leaf spot and all oilseed rape diseases. And then I'll be passing over to Dr. Faye Ritchie from ADAS, who's going to talk about management strategies. And we'll then open up into a panel discussion. And Adam Christie from Scottish Agronomy will be joining us, as will Rachel Wells from John Innes Centre, who'll give some insight into the precision breeding aspect of the project. We'll then open it up to a Q&A, and you'll see at the bottom of your screen there is a chat and a Q&A. So if you've got any questions that you want to ask, then please do put them into the Q&A there, and we'll aim to get to them at the end of the webinar. And then it's due to close at about 9:30. So just for those of you who don't know Boffin, we started about 5.5 years ago to represent farmers who do on-farm trials. We have well over 3,500 members with almost 750 farming innovators. Now, within that, we have over 70 active trialists now, most of whom are delivering our 5 major DEFRA-funded projects. We are adopt facilitators. We have 6 farmer-led projects underway and more in the pipeline. So if you're considering adopt, do get in touch and we can guide you through it. And look out for Launchpad, our new initiative that connects agri-tech innovators and farmers to develop, test and launch products through on-farm trials, ensuring that innovators solve genuine farming challenges. So we aim to put farmers in the driving seat of innovation by connecting them with science, supporting on-farm research and sharing knowledge. We're intensely collaborative, working closely with scientists and tech innovators. Knowledge has value, but only when it's shared, and we provide the platforms to do that. So farmers can progress, push the boundaries of productivity, and take their rightful place at the forefront of the fourth agricultural revolution. So the major projects that we're involved with are on slugs with Slimers, which has developed new slug prediction maps and progressed automated detection and novel forms of control. Truth is our soil and root health project, gathering a staggering array of data for our trialist farmers on root health and the soil microbiome. In NCS, over 30 Pulse pioneers are developing ways to improve performance from peas and beans, while Probity is bringing precision-bred cereals to commercial farms for the first time across Europe. But today is all about LLS Erased, light leaf spot enhancing resistance and reducing susceptibility with editing. LLS Erased brings farmers and scientists together to tackle the growing threat of light leaf spot in oilseed rape by advancing and testing precision-bred traits and developing a new disease management tool and integrated pest management strategy. So it's led by Boffin. It's a 3-year, 2.5 million multi-partner project. You can see the other partners involved at the bottom of the screen there. It's funded by DEFRA's Farming Futures R&D Fund, and that's delivered by Innovate UK. Now, at its peak, just over a decade ago, actually, the UK oilseed crop stood at 700,000 hectares. We were the 5th biggest exporter of rapeseed in the world. Today, best estimates suggest that area has plummeted by almost 2/3. We now import 20% of our edible oils at a cost of £1 billion to the UK economy. Now, while crop damage from cabbage stem flea beetle due to the ban on neonics led to the crop's demise, in recent years it's been light leaf spot that has posed an increasing threat to the crop that remains the most widely grown break crop in the UK. Now, crop losses caused by light leaf spot peaked at over 300 million in 2022. It's oilseed rape's #1 disease, and the problem with the pathogen responsible is that it cycles several times in a season, unseen until it's already taken hold, often rendering fungicide applications ineffective. Control of initial infection is vital, but there's no risk forecasting system to guide growers' timing of fungicide applications. Now, while breeders have improved host plant resistance, populations of this pathogen shift and new virulent strains that overcome genetic resistance. So we need to bring this crop back. The industry calls for a reboot, but until its disease weakness is addressed and mastered, this is a futile aim. Now, precision breeding by gene editing, however, offers new opportunities. Scientists working on the project have recently identified a novel susceptibility gene that could prove to be a game changer. Unlike traditional gene for gene interactions, in which a mutation in the pathogen population can break down resistance, they've introduced a specific change in the plant's genome that has conferred considerably reduced susceptibility. The pathogen simply finds no place in our novel plants where it can take hold and cause its devastation. But this isn't just about relying on the trait. We will develop a new risk forecasting to give growers real-time decision making by monitoring the emergence of new virulent strains in farmer-led trials. We've developed molecular markers for rapid detection of pathogen race structure, and you'll hear more about that from Yongju in a bit. And a new decision support tool for in-season fungicide application guidance will bring growers effective disease control that is truly risk-based and data-driven. But that is just the start. What lies at the heart of our approach is a new grower-led platform to share skills and resources and foster understanding. Our vision is a farmer-led field trials providing a pipeline of precision-bred oilseed rape traits to the industry. We are working with industry partners to develop understanding of how to tackle this tricky pathogen, and we've set up a new OSR Circle community, and that's where we can share our understanding of how to drive productivity in oilseed rape. Now, one huge strength, and this is one of the bits I'm really excited about, by the way, of this project is its collaboration with international partners. And a really exciting aspect is the involvement of CBUS, who are based in San Diego in the States, with their RTDS system that introduces editing tools free of transgenes. These simply aren't in commercial use yet anywhere in Europe, but they're coming first into the LLS Erased platform. Combine this with the breeders bringing into the UK pre-breeding expertise from their EU parent companies. It's an unprecedented level of talent and resources to develop a pipeline of traits, a wealth of strategies right here in the UK that will benefit farmers across Europe and further afield. And we're looking for growers to get involved as well. So now I'm going to hand over to Yongju shortly. But before I do, please get your phones out and scan that QR code. Because we're on the hunt for spore scouts. Now, Yongju will explain more about the pathogen populations and how they vary across the UK. But if we're to fully understand this, we need samples from all across the UK. So we're asking farmers and agronomists to find infected leaves and send them in for analysis. We can provide sampling packs with postage paid envelopes along with guidance on what to look for. But that pathogen, it's cycling in your crops as we speak, and we need it. We need those pathogens, and we need to be able to learn more about them. So we're going to be running on-farm trials from this autumn too, and we'll shortly be on the lookout for trait trialists who will be paid to work with us to develop new ways to monitor the effect of disease in crops. Those same trait trialists towards the end of the project will be the first across Europe to get the chance to grow precision-bred oilseed rape. So this is a really exciting project, and you can get involved today. Sign up to be a Spore Scout, join the OSR Circle. But look, I've whittled on enough, so it's time to bring in the real experts. So Yongju, if you'd like to join us.
Hello, good morning, everybody. So I'm Yongju Fang from University of Hertfordshire. So I'm just to talk a little bit more about lettuce sport. Yeah, click. Yeah, so I just gave introduction about lettuce sport. Is now become a major disease on oilseed rape in the UK. It's taken over former stem canker now. And this disease is caused by a fungal pathogen, Piranha piscina, brassica, and it can infect all parts of oilseed rape and also other brassica crops. So the typical symptoms Yeah, go back, back to another one. Back, Tom. Yeah. So the typical symptoms of left spot is on the leaf. You can see that it's like a circle with the a white dots is the asexual spores, and then also infected stems and pods. The main ear loss is caused is by the pods. If the infected pods get shattered, so you get the ear losses. So next slide. Yeah, night spot is not all disease on earth rape and also other breast care crops, especially you see in the UK and the kale. You can see the typical symptoms on the kale. And in the front of the leaf and the back of the leaf, you can see the sporulation. Sporulation is producing the asexual spores. This can spread to the other part of the plant and also nearby plants as well. And also, if you observe some of your vegetables bought from supermarket, you can see the lettuce spot on the Brussels sprouts as well. And also, you can get also cauliflower and broccoli as well. So it's not only disease on earth rape, but also on other breast crops. Next slide. So the life cycle of the pathogen. So the epidemic started in the autumn by the sexual spores, ascospores produced on the crop debris. So the ascospores released from epithelium and then landed on the leaf surface. The germinated ascospores can directly penetrate the leaf cuticle and grow in the subcuticle spaces without any symptoms until they accumulated enough biomass and then the asexual sporulation and then they break through the leaf surface so you can see the symptoms. So once the symptoms are observed, this is the you can see the white dots and like a sort of like dots. This is the asexual fruiting body and they contain lots of candida. They can spread by rain splash to other parts of the plants, so they cause the secondary infection. Furthermore, after the infected leaves dropped off in the field, they also can produce sexual spores again and then release excess spores and also infect the plants again. So within the one crop season, the pathogen can have several life cycles. Therefore, this disease is a polycyclic disease. So it's very difficult to control. Next slide. Yeah, so this is the typical symptoms in the field. Normally you can't see very severe, not very severe, because the disease have a symptomless period. Normally infection occurred in autumn, but the symptoms are not visible until spring. For our do research, if you can't see the symptom in the leaf, you see it's not, for example, the leaf A is not severe. However, when you incubate it to promote the asexual production, you can see actually this leaf is infected very severely. You can click again. Yeah, you can see it now. So this is the same leaf in the field, and also after incubation at the lower temperature for 5 days, you can see the leaf is full of spores. So it's severely infected. Because it's a similar period, also another layer making the control of left spot is more difficult. Next slide. Yeah, so how to control the disease effectively? So we need the two methods. One, we need to understand the host resistance, so we can use the cultivar resistance to control the disease. Secondly, we need to understand the patient population, so we can deploy cultivars with an effective gene in that area, because the patient population can evolve to overcome the host resistance. Next slide. Yeah, so host resistance, currently we have two types of resistance to control this disease. One is major gene mediated resistance. Second is quantitative resistance. The major gene mediated resistance is mainly to stop the secondary infection because they don't produce a Candida spore. And quantitative resistance, they only reduce the sporulation, but they only reduce the number of sporulation, reduce the spores to then reduce the secondary infection. But it's not completely controlled. But if you have really effective resistance, there's no sporulation. That means no secondary infection. So control this disease, we rely on these two type of resistance. The major purpose is to stop or prevent the secondary infection. So to control the disease at the beginning of the season. Next slide. So you can see in the field the severe infection of the leaf. For example, on the leaf, the first photo, you can see the severe net leaf spot on the leaf. After by rain splash, the spores can spread onto the nearby, onto the other part, onto the stem of the plant, and also spreading on other plants as well. Under this under the suitable conditions, you can see the sporulation of the pathogen. You see on the stem. Another slide, you can see the white dot is the asexual fruiting body. So because if these stems in the upper stems, and they can later on after rain and rain splash and the wind, they spread into the pods and the flower buds. It's causing major air losses. Next slide. So currently, we don't have the information about the pathogen populations. We don't know the pathogen races for effective use of the cultivar recent. For example, we have the cultivar flamingo, AHDB recommended list for 2017-18 is resistant for lettuce spot. The resistant rating is 7. However, we grew this. We have field trial in two sites. One is in Herefordshire. Another one is in Cambridgeshire. You can see when the same cultivar grew in the two different locations. In Cambridgeshire is resistance, no disease. However, in Herefordshire, they got severe disease. So that means the pathogen populations in the two sites are different, and then the pathogen population in the Herefordshire they may be mutated to overcome the resistance. So let's lead to understand the pathogen populations, so we can deploy cultivars in regions where the environment race of the pathogen is a predominant. So yeah, click slides. Yeah, so this come to the one important work we do in this project. Next slide. So this project we will understand. We need to understand the pathogen populations for effective use control of the lettuce spot, use cultivar resistance. So firstly, we need to detect the pathogen races from leaf samples. So to understand what the pathogen population in the field conditions. To do this, we need to take leaf samples from different regions. So this is the help. We need your help to take samples from your fields. So once we got the leaf samples, we will isolate pathogens from the infected leaves, and then test variation of the pathogen virulence between different regions. So with this information, so we will have an idea which cultivars will be effective in which region. And secondly, secondly, because net leaf spot, the epidemics started from airborne Asch's spores. So we another time, so we need to monitor the timing of the pathogen spore release. To do this, we will set up spore samplers also in different regions to detect the timing of the sports release and abundance of sports release. So this will help to develop management tools to guide the fungicide application. So make the application is more targeted and more effective. So yeah, I finished now. Thank you.
That's great. Thank you so much, Yongju, for that, giving us an insight into the disease. I have to say, it's one of those things. It does lurk silently in the field, I think. But we'll move straight on to do get your questions into the Q&A if you've got any questions about the disease or anything like that, anything that Yongju has mentioned. Do put your questions into the Q&A. But let's pass straight over to Faye.
Thanks, Tom. Good morning, everyone. So for our part of the project and for many of the people on the call today involved in the project, in fact, everybody, we'll be also looking to improve light leaf spot control using integrated disease management strategies. So next slide, please. And there's always the question, well, why should we focus on light leaf spot? And Tom's mentioned it and Yongju's mentioned it. It has become increasingly difficult to control. I mean, 15 years ago, we might see 90% of control with fungicides, but we more routinely see about 50%. That doesn't mean they're not doing anything, but there is opportunities to improve this substantially. The yield losses do exceed other diseases now. We know that there are azole insensitive strains out there, so there is that insensitivity to fungicides. It's not impacting field performance, but we should always be bearing that in mind when we're thinking about control measures. And I think we should recognise that conventional breeding has made substantial improvements. If we look at the recommended list varieties, so I've just put the hybrid and the conventional varieties on there, the top right, you can see that a lot of the varieties now have a resistance rating of 7. So that's good resistance, but there are still benefits from applying fungicides to those varieties. And just to demonstrate what the value of variety resistance is in terms of disease management, I've put another little chart underneath there that just shows the difference in the epidemic development across November into January on a variety with a rating of 5 versus a variety with a rating of 7. So you can see that, using that more resistance and that good resistance substantially reduces that disease development. And when it comes down to it, integrated strategies are more likely to be more sustainable and improve the effectiveness of disease control in the long term. Next slide, please. So we need an integrated approach to crop protection because pathogens will just continue to overcome control measures that we use against them. No matter what we throw at them, they're going to do it. But we do have limited trials data where we investigate integrating varieties and fungicides. But the trials we have done do suggest that inputs could be better targeted. So on the right, this is some data from a trial we did with KWS over 10 years ago now, and we've shown it many times before. And what you've got on the left is a variety with a rating of 7, so high, higher resistance, and a variety with a rating of 4 on the right. And you can see that the blue bar there, that first bar, just by using a variety with good light leaf spot resistance, gave you a yield response of 0.35 tonnes a hectare. Now, the other thing to bear in mind with this data set was it wasn't a particularly high disease year. So actually the benefits from using varieties with better resistance are potentially much, much higher. We also looked at a number of fungicide programs. And you can see the next bar along where there was sprays applied in November and March. You got a small increase in yield, but it was the last 2 bars where the fungicide programs were more effective. And what we found there was it was the December application that was most effective. So not the sort of traditional November spray or the spring spray. And there are challenges around December applications because in many cases people can't do them. So if we had a better forecast tool, we could make those decisions. We could say there's no point in spraying in November. The best time will be December, but actually we can't spray, so there's nothing we can do about that. And the same with the later sprays. So there's real opportunities, I think, to fine-tune how we manage diseases, especially like leaf spot. Going forward, we're going to have fewer chemical modes of action available for disease control as well. And we're also going to have to think about testing non-chemical alternatives, and those are going to need to be targeted effectively as well. So what we understand at the moment is that fungicides can provide additional yield, even on varieties with good resistance, but we need to make sure that those inputs are cost effective. Next slide, please. So I think this is a really exciting part of the project because we've got the opportunity to develop real-time forecasts and use local weather data and really enhance the way that we can predict risk. So many of you will be aware of the previous forecast that was developed, and I've got an image of it at the top right there, where the risk was assessed by region. And of course, that varied from year to year, but it was very much at that higher level. So as part of the new project, what we're going to do is we're going to review all the information out there, previous, current guidance, get all those metrics around disease risk and build a combination epidemiological and weather-based model. So bringing together what we understand about the life cycle of the pathogen, that Yongju went through. So we'll need to consider things like spore release timing, and the populations as well, and build that in so that we can predict the risk and potentially the inputs that are needed to give you that additional yield. But I think the key part of it, and one of the most important things within this project, is testing it and making sure it's fit for purpose. And I think with everybody getting involved and providing their input into how we've done it and how they think could improve it is going to be the most important part. It's because we need to make those model outputs fit for purpose. And at the moment, we're thinking a good place for it would be the IPM decisions platform. So that was built as part of a Horizon project and considered to be a sort of one-stop shop for decision support tools. So if we do that, that would make it available and free to all. But of course, we will consult with everybody to see what they think about that and how we would do that. Next slide, please. Another important part is testing integrated pest management strategies, in this case, disease management strategies on on-farm experiments. So many of you will be aware of agronomics and using tramline trials. And what we want to do is look to see if it's feasible to include novel experimental designs that will help people to understand and predict how these IPM strategies will perform on their farm. So for example, you could run a trial and you can predict the outputs of a number of the other treatments. So the kind of question you could ask in this type of trial in relation to this project would be, is it cost effective to switch from this variety A to variety B? And do I need to treat it the same as I would for my previous variety, for example, with a two-spray program? So we're investigating that because I think it's really important to understand whether these changes have the potential to reduce fungicide spend applications, but specifically on farm and not just in small plot field trials. Next slide, please. So overall, what will the project outputs be? A key part will be disease management information to support the use of gene-edited or precision-bred varieties. But the other thing to bear in mind is it will also be relevant to all the current varieties out there as well. So it's not something we have to wait to use. We can use it now if we need that. There will be a decision support tool that uses local weather data to help guide fungicide decisions. So, there's no point in spraying here, but actually it would be better to do it here. That sort of thing, I think, will really help improve the yields, especially if we want to target those things better. And if we do that in relation to disease risk, that's the key thing there. We'll have methods to help test gene-edited or precision-bred varieties on farm as part of integrated disease management strategies. And I think having that local information is really, really important because, as Yongju explained before, there's differences in populations. There's going to be underlying differences on different farms. So we need that local information. And if you take all of that together or even parts of it, these approaches could be applied to any disease, any trait, any crop and input. So it would be nice to sort of use this as an exemplar to get the methods up and running, but actually the potential is to use them on a different scale as well. And next slide, please. And that's everything. Thank you.
Great. Thank you. Sorry about that. Thank you very much. That's great. Well, look, that was a really fascinating insight into the disease and the management strategies around it. So I'm going to ask our experts to join us. So Yongju and Faye, if you'd like to switch your cameras on, and then also we have Rachel Wells and Adam Christie to join us as well. So Rachel is a leading scientist at the John Innes Centre. Many of you will know the work that she's been doing into osteohydrate for many years. Delighted to have you with us, and delighted to have you on the project, Rachel, as well. And Adam Christie is leads the Scottish agronomy in Scotland as well. So great to have you here today. So I was just going to ask, first of all, Rachel, I briefly described the precision bred element of this project. Do you want to just give us a little bit of an insight into the really quite groundbreaking technology that you're going to be using through this project?
Tom, I mean, I think this is a really great opportunity to start to look at the power of precision breeding and actually what it can do to us. So in this project, I'd just like to say, though, we're also taking forward some conventional material. So we have standard material that's been produced by mutation breeding, but also then we've got that shortcut of using precision breeding to produce those precise edits in the genome. And the important thing with regards to comparing that to our conventional mutated material is the fact that we're looking at doing that in cultivars that are more adapted to the UK growing environment. So stuff that's, more close to market than our traditional populations that we use for our mutation breeding. So we were very lucky in that when we were screening for resistance to light leaf spot with the University of Hertfordshire, we identified this new type of gene. And it's, we've always relied on pulling through resistance genes to provide crop protection. But actually, a lot of the interest now in the field is in reducing this susceptibility to the pathogen. So these genes work in different ways to our standard resistance genes. So we hope there's going to be that greater longevity of resistance. So because we found this gene that is linked to susceptibility, it means we can go into that genome and we can just basically take out the function of that gene. It's not essential for the plant, but it is essential for the pathogen to be able to come in and infect. So what we've done is we've gone in and we've edited that gene to remove the function, okay, to knock out that gene so the product is no longer produced within the plant. Within the project, what we want to do is trial how that affects the resistance. We know it increases resistance in the mutants. We need to go through and show that within this precision bred material. But then also we want to know how our material, both our conventionally bred and our precision bred organisms, behave on farm. Because it's one thing to show we've got resistance in the lab when we're using our lab strains of the pathogen, but we need to show how that interacts across the UK. Now, in getting this material out, it's great because it's the first precision bred material we've got that's going out into the breeders' trials and then finally out onto farm. But really, it will provide us with the mechanism and the knowledge for how we need to make our stakeholders happy that this material is suitable, and the process we need to go through with the regulatory bodies and the food standards agencies, for example, to be able to bring this into part of the UK supply chain.
Great. Now, one aspect that I touched on as well was the involvement of CBUS and this rapid trait development system that they've got there. Do you want to just briefly describe what that is? What's different about this that is perhaps different to the CRISPR-Cas9 technology that many of our farmers will have heard about?
Yeah. So conventionally, when we're doing developing a precision bred organism in the lab, we'll go through a transformation, so a standard GM approach, and then we will take away the construct that we've used to introduce that edit. Okay. We have to do that during the process of regenerating plants and selection through breeding. Now, with Cebus, they approach this slightly differently. So they work very closely with the breeders. So for a start, they're working with material that's directly relevant to the breeders. So they'll take a breeder's cultivar and they'll test it for suitability for use in their approach. They then perform everything. They develop protoplasts. So basically, these individual cells. So rather than transforming a whole plant and going through regeneration, actually, they're going through working with a whole cell, a single cell. And then develop plants that are transgene free from these individual protoplasts. So actually, you never get a plant developed that is GM and have to take away the GM part. You're clean from the very beginning. And they can do this incredibly rapidly. So to go from individual protoplasts to your precision bred organism, actually the first seed's produced within 12 months. So I think it's really exciting for like kind of developing that pipeline. Once we know what genes we're interested in, it then becomes a very simple process to push that through into breeders' material.
No, that's really exciting. I mean, that really compresses. I mean, it basically sort of pushes about 10 years worth of traditional breeding into one or two. It's very exciting. But I want to bring in Adam now. Adam, as you know, a large part of the campaign that we've got, and we're certainly launching today, is the Spore Scout campaign, and we're encouraging people to find leaf samples and send them in. Now, as Yongju explained, it's an incredibly difficult disease to spot in the field. Now, your agronomists have obviously, been spotting it in the field for years, and I'm sure you've got experience of sending samples in. What would you say that will help those people who are interested in becoming Spore Scouts? What sort of advice would you have for them?
Don't be scared. Get out there. When you find samples, get them in. You will benefit directly from sending those samples in. It's an incredibly tricky challenge like leaf spot, and I always feel we fight with one hand tied behind our back. If you read the literature, it will normally tell you that it's only like leaf spot if you see active sporulation. Well, I can tell you we've a wind chill of about -3 outside at the moment, and there's not much active sporulation going on in the field right now. But we had some of our trialists scoring like leaf spot at 10 to 15% leaf infection last week. So, I mean, the pictures that Yongju put up were very good in that they showed the disease in its many different forms. Like leaf spot likes to disguise itself and hide under many guises. But basically, there's some very good information online on how to identify the disease. And if you're unsure, stick it in a poly bag and put it in your airing cupboard for 24 or 48 hours. That will start it sporulating. Once you've seen that, go and take another fresh sample and send it in. But to me, part of the success of this project will be delivered by actually getting to know our enemy and finding out what races are where. And our experience of light leaf spot in Scotland can be very different to the experience down south. But we're a generous race, the Scots, and I firmly believe that we are sharing our strains of light leaf spot and sending them further south. So we might be the harbinger for the disease, but it's chances are it's coming your way.
Yeah, no, thanks a lot, Adam. No, that's great to hear. No, that's super. Look, I want to turn to Yongju. Now, we heard from Adam about the difficulty sometimes in finding it in the field. So again, just thinking about our Spore Scouts going out and looking in the field. The problem, I think, as you described very clearly, is that it's a really, really difficult disease to spot. So when you're going into the field, what are the sort of signs, what are the first signs that you look for that you think, aha, there could be an infection here?
Okay, yeah. So this pathogen also because they interfere with the plant hormone, so they can sometimes once the infection they cause the formation of the plant change. Sometimes you can see the leaf tender kernel is not normal leaf is very flat, and you can see the leaf kernel is one of the symptoms. Another symptom you can see is the plant if you notice the infected plant is stunted and is especially severely infected plants is shorter than the normal plants is stunted. So this is for severe ones. For the leaf also you can see the leaf curly and even without seeing the symptoms you can see the leaf is not very flat. It look like it's crispy. They are less curly a little bit. This is one of the symptoms. And
Right.
also, it's more easy to spot the damage is after the frost or after cold. You can say when the cold is normally promote the symptoms development. So normally you can go to your field after a cold period. You can see the symptoms.
Great. I want to come back to Adam because the one of the things that I get confused about is that I've always understood light leaf spot to be a spring disease, and that's the time that it's causing it. But we heard from both Faye and Yongju that it's actually in the autumn. So what's your take on this, Adam? I mean, because we're encouraging people to look for leaf samples now. But should this really have started back in the autumn? I mean, is autumn the more important time, or is now the time to find to find those infections and to deal with them?
There's absolutely no doubt the disease is present, but it's often at subcutaneous stage, and we don't actually see a huge amount of expression in the autumn unless we tend to get a very open, mild autumn. So the biggest issue would be you could have the disease and not actually be aware that the disease is in your crop, and expression will only tend to come either through prolonged frost, where you get leaf scorch symptoms, or when you have a period of warmer weather where suddenly sporulation occurs. So the reality is the disease is there, but you don't know it's there until it's almost too late. I mean, traditionally, we would have always sprayed in the autumn to control the disease. The challenge we found with that spray is you were using a single active in, say, October or November and expecting it to control disease for 3, 4 months during perfect conditions for that disease. The upshot being that you ended up selecting a population that was resistant to your main fungicide. And then to make matters worse, we went back in at stem extension with the same product. So it's little surprise that we have an issue with the disease, and that's why for northern growers, variety resistance is our key control method for the disease now. But yeah, it's only really in the last 3, 4 weeks that we've had visual symptoms to be able to confirm that the disease is there. And this is quite a high pressure year. We're seeing a lot of disease out there, and it's very widespread amongst our fields at the moment.
Right. So presumably there's going to be quite a bit of infection out in the field, even if you didn't know it, and perhaps getting out into the field, really digging down into your crop and looking for those infections will discover those infected leaves, which of course we can collect and send in for sampling and give us a much clearer idea of the disease, I guess.
Yeah, I mean, this mapping of the population is
Yeah.
going to be absolutely essential. We,
Mm.
the first rule is always know your enemy, and we do not know enough about this disease. I mean, certainly in a Scottish situation, the chemistry is no longer a reliable partner. We've got to find other methods of staying on top of the disease because the yield loss potential is massive. And I mean, we're talking about a crop that's one of the few bright spots in arable agriculture at the moment. I mean, growers can effectively lock into a profit if they successfully grow oilseed rape. there's not many other crops that are offering that potential at the moment. So it's absolutely the right time to be doing this work because the headlines for oilseed rape have been about other issues, either to do with, poor establishment with drought, flea beetle, cabbage stem weevil, and such like. Light leaf spot's never gone away. It's a never-present challenge for us that, along with clubroot for northern growers, is probably the greatest challenge that we face in successful oilseed rape production.
Now, Faye, Adam talked about the problems with using fungicides in disease control, and you talked about it as well in your presentation and the disease management tool that you're developing within the project. So just talk us through that a bit more in terms of where are the fungicide strategies failing? What are growers doing wrong that we need to, refine, improve?
Well, the first thing is, I don't think growers are doing anything wrong. I think we have a set of guidance that was developed back in the sort of 1990s, and we've got a pathogen population that's continually evolving. And I think we've got technology now that can help us be more precise and make decisions that are cost effective. The traditional timings for like leaf spot sprays, October, November, and another spray, sort of stem extension, pre-stem extension in some cases, and sometimes on the pods. But I think we can really now use technology to fine tune that. I think the populations is really, really interesting, and I'll be really interested to see what's going on in Scotland because we've got that, we know there's that difference between Cambridgeshire and Herefordshire. So what's that? What do we the population understanding that is really key. And I think if we can find a way of better targeting fungicides, we can, there is potential to improve yields with fungicides.
Hmm.
And
Yeah,
we need to just understand how to do that better. And I think if we can develop a disease tool that can help guide those decisions, then it'll be a big step forward, really, from just the traditional spray times.
yeah. Now you mentioned this Horizons project and the disease management system that's been developed through that. Why hasn't that been progressed into a tool that growers can use?
The Horizon project, it just ended a year or two ago, and effectively what it is, it's a platform where you can store decision support tools. So it's almost, I mean, it was sort of badged as a one-stop shop for decision support models. So the vision was that this was the place people could go to make, help guide their decisions in relation to disease management and things. So I think it's there now, it's ready to be used. So I think let's see how we can integrate this into that and start using it, because I think having all this information in one place is going to be key to helping people to use these tools.
And that's one of the aims of the project, I believe, isn't it? To bring it from it's great to hear that that research project has taken place and that the ability is there to do this DMS. Let's go ahead and actually do it, which is exactly what we're going to be doing through this project, I think, isn't it?
Yeah, I mean, to be able to build a model and a forecast tool and put it on that platform and make it available to everybody, I think is a really important part of this project.
Yeah. And I was just one other thing I wanted to ask is that so a lot of the farmers on the call will be familiar with the likes of Zavio, FieldView, and those sorts of systems. Syngenta has one as well. Why haven't they stepped up and delivered these DMSs? Why does it have to be a taxpayer funded project that does this?
I think that's a really good question. And in my opinion, I think, for something to be viable, something has to be, you have to make money out of something to develop it. And there's not really anybody, a big marketplace to sell decision support tools into. So being able to create these using government money and making them available to everybody, I think is the real value. There's always going to be things that commercially won't be funded by businesses to develop because, I mean, in this project, it's, Yongju and University of Hertfordshire who are doing the model for that part of the project. So that's very much done, through sort of academic routes with input from us from the point of view of data and the sort of things that can help do that. But ultimately, I think there are things that can really support growers that fall down a bit of a hole in terms of commercially, these can't be funded. But if we've got government funding, we can do this, and they'll make huge improvements for growers.
Yeah, I mean, it has to be said, of course, if you look at the likes of BASF and Bayer and so on, they're sitting on global budgets, but global markets as well. Why on earth should they have to spend the money developing a tool for a disease that is not only sort of specific to the UK, but it's very, very specific, as we've just been talking about, to different regions within the UK. And so, to develop a tool, it's not an easy business, and that's why I think it's going to be really interesting as we develop this, and also as we see what the pathogens are doing in the field, and we get into the trials, which will be going on later this year. I mean, just talking about the trials. Now you've got a particular method that you're looking. You talked about agronomics, which is the ADAS system of assessing farmer-led trials. There's a new method that you're developing. Do you want to tell us a little bit more about that? Because that's something that I'm really looking forward to, I have to say.
So I think Susie's on the call actually. So Susie Rock at ADAS and myself have been looking at bringing a method called the Taguchi method into tramline trials. And what that allows you to do is to test a sort of small number of treatments in the field and predict how a larger set would work. So in small plot trials, we can run, say, a 16 treatment trial, and we can get thousands of different combinations. We can work out how, from field data, we can predict how those will work, thousands of combinations. For tramline trials, we're a little bit more limited, but what it would allow us to do is to do a sort of standard 4 treatment tramline trial and predict the outputs for 8 treatments. So it just allows us to understand more about, so for example, the example I gave was, I might want to switch variety. Let's do a trial and put that variety in this tramline trial, and I'll do everything. I'll put my 2 sprays on and see what happens. And you can get an indication of how that would work with your standard practice. And also, in that particular design, it would say variety A versus variety B. Here's it with nothing applied. Here's it with a spray in the autumn. Here's it with a spray in the spring, and here's it with both sprays. So you can predict how that will turn out, and I think that would be quite a useful piece of information to have.
And just to be clear, I mean, this method, I don't think it's used. Is it used elsewhere, or is this the first time it's
used predominantly in engineering? We not so much in crop protection, but it has been used to understand fertilizer inputs.
Right.
You can have up to 12 factors in a trial. You can look at a lot of things at the same time. So I think it's quite an exciting way of getting rid of the things that won't work and focusing on the things that actually will, and then testing them on farm.
Great. Well, that's going to be a really exciting element of this project. By the way, I'm conscious that I've been hogging the question. Now, we haven't got any questions coming through, so I don't know whether there are people who perhaps are holding back. But please do come through with your questions and put them into the Q&A, and we'll address them to the experts here. But I just wanted to ask you, Rachel, come back to you. I mean, if we've got this, precision bred, reduced susceptibility, that you've developed, then why do we need to develop all the decision management tools and so on that we were talking about with Faye? Why do we need to look in such detail at the pathogen population if we've got this miraculous cure through precision breed, or solution through precision breeding anyway?
I think we have to take knowledge from the past and what's happened with the breakdown of disease resistance. I mean, we're trying to farm smarter, we're trying to farm with less inputs. By using the knowledge we've got out there, we can actually tailor that solution. When you look at integrated pest management, it's not one thing. This is the whole thing. It's integrated. It may start at the bottom of that IPM triangle with resistance, but actually it takes a layering of all those different techniques to give you that effective control with reduced inputs. So,
Great.
I think it would be very foolish to say that any disease resistance is going to be that silver bullet to provide a solution to a disease. We know disease resistance can be overcome in other systems. So I think it's something we have to look at protecting going forward and farming in the best possible way using everything at our disposal.
We've got a question come through on the Q&A from Julian Little. Thank you very much for your question, Julian. So, do the seed breeders get involved at the end, or are they integrated into the program from the start? Can I ask you to answer that one again, Rachel?
Yeah. So the breeders are in here from the very start. When we originally identified the susceptibility locus, that was in collaboration with a breeding partner. As we go forward, their inputs are really, really important for how we get this precision bred material to the field, because we need uptake from the breeders to actually go forward with this, and then uptake from the whole supply chain so we know we've got a market to pull that product through. So to start with, we're going to be working very closely as soon as we've bulked up enough seed with the breeders to put this up into commercial scale seed bulking lots, and also at the same time doing those initial disease resistance trials alongside with the breeders as a first look-see with this material.
Great. Now, Penny, thank you very much for your comment in the chat there. Penny Hundlebury from John Innes Centre, talking about we have the science, we have the enabling legislation, but projects like this are vital to look at all the challenges to get to the market. Now, I know from the experience with the Probity project, there are still many challenges. I mean, as Penny points out, we have the enabling legislation. Now, that is an element of this project too, isn't it, Rachel? Looking at finding our way through that legislation as well. I mean, what are some of the challenges that still remain, and how do you hope that we're going to be getting over those?
Yeah, so I mean, now with the enabling legislation, it's relatively simple to get this material on farm. But you need to look beyond there at bringing this into, we need to go to the seed sales, actually. We need to go to the crushers, and we need to go to the end consumer. And it's understanding the challenges that we'll face at each part of that supply chain, especially in our interactions with Europe, and within the interactions within the UK. We know legislation is not the same across the UK market. So, it's giving us that opportunity to develop that understanding, interact with the stakeholders, get feedback on what we're doing, but also what we're required to do to effectively bring this to market. And this is not just for light leaf spot. We're seeing this as an exemplar project for all those other traits that we're currently working on in research that we would like to bring through.
Yeah, that's one question. I know we've gone slightly over, but that's one question I was going to ask Rachel, and I know that a lot of people on the call are going to be thinking about it. How close are we to cabbage stem flea beetle?
So, we're nearly coming to the end of the work we were doing, or the project we were doing on cabbage stem flea beetle. We've had some really nice outputs from that project. For larval cabbage stem flea beetle in Brassica, it's really not a good tale at present, because in all the varieties that we've screened, we haven't found the resistance that is there to the larval stage. For the adults, it's slightly more promising, and yes, we could see that resistance to the adult feeding was present. So we also know that resistance, we'd identified a region of the genome that is linked to susceptibility. So very much like this light leaf spot tail, we have a similar tale potentially with cabbage stem flea beetle. So we're still not there. We've got it down to a region of the genome. We still haven't got the causal gene. But I have a student at the moment that's going on and testing our best candidates. So hopefully, we should be able to make some progress there and actually start to pull that through. We've
How? Yeah, sorry.
On that project, Tom, we've opened all those results up to the breeders that are involved. So actually, they've had access to all the material we've been working with. That in their selection trials as well. So I expect we're going to see something hopefully come through very soon. We already know that if you look at the material that's out there on the recommended list, it's more flea beetle resistant than what we had back in 2013. So I think, we're making promising step forward.
Great. Oh, that's great to hear. And I guess, you will hear about it here first. So listen, I just want to say thank you so much to Rachel, to Faye, to Adam, and to Yongju for joining us on this. And I'm just going to bring up my screen again and share if hopefully it'll work this time, and share no, it hasn't worked. Wait a minute. Let's try that again. Uh, there we go. There's that QR code to sign up to be a Spore Scout. Do please join. Also join the OSR Circle. You will get to hear about all of the advances through the project first on the OSR Circle. That's our new community for oilseed rape growers, to share knowledge and experience. We'll be posting updates on how things are going, and anything to do with the project, including what Rachel's been working on in other aspects that she's just touched on there as well. So please do follow the project. Please do join as a Spore Scout. Get those samples coming in. We've got packs ready to go out to farmers, which will give farmers and agronomists, which will give you full guidance as well. But obviously, if there's any questions or queries, then you can come to us through the OSR Circle, and we'll help you get a grasp on this really tricky and difficult disease. So look, thank you so much for joining us. I'm sorry we've gone slightly over, but I hope you enjoy the rest of your day, and goodbye.